Hi, it's Professor Tiffany Martin with Medical Microbiology, and we are continuing in Chapter 7, Microbial Genetics. And we are down to, well, we've talked about the structure of DNA. Now we're going to talk about what's the DNA's job. What is the role of DNA? And DNA actually has two roles, and both of them are equally important. Most of the time we think about making proteins, the instructions on how to make a protein, and that's true. That's called gene expression, very important. But equally important is DNA's role in DNA replication. It needs to be able to copy itself. Remember, they were talking about bacteria and they are passing down their genetics. They're copying that DNA and passing it down to their progeny. Being able to make an accurate copy is extremely important. You do not want mistakes in your instruction manual. And you can go back to Grandma's recipe book. And so Grandma has her recipe book, and when the time comes, poor Grandma, it's time to pass that down to their children and grandchildren. Say grandma has two granddaughters and they both want the recipe book, then it is very important to be able to copy that recipe book so that both of those grandchildren have an accurate copy. Otherwise, they won't be able to make those chocolate chip cookies for their family and pass that recipe down. So DNA replication is very important. So let's go to our chart. Two rules of DNA: replication, so we need that accurate copy so that when we go from one cell with one chromosome to two cells, we have each cell has an accurate copy. So that is the first role of DNA. The second role of DNA is going to be that gene expression. And when we talk about that, the DNA is the instruction manual, but it doesn't do anything other than provide the instructions. If I want to actually taste grandma's chocolate chip cookies, well, then I got to get out the flour and the chocolate chips and everything needed to make those cookies. I've got to express it. I've got to make the recipe. And that ends up with the product, the chocolate chip cookie, the protein. This is called gene expression. And gene expression always in all organisms goes from DNA to an RNA intermediate to a protein. So this is a very old process because prokaryotes do it basically the same way eukaryotes do it. So again, like metabolism and cellular respiration, this is a very old process and used by many different organisms. So the things you learn here, while they're geared to a prokaryotic cell, are going to lay down very nicely on things you've learned in eukaryotic gene expression as well. All right, so DNA to RNA to protein. And again, because this is so universal, this is called the central dogma. So DNA to RNA and protein is referred to as the central dogma, and this makes up gene expression. Okay, so DNA goes through an RNA intermediate, and now the type of RNA that gets translated into a protein is actually referred to as messenger RNA. This is a gene that is going to get transcribed, it's called transcription, into a strand of RNA referred to as messenger RNA. And that messenger RNA is going to go to a ribosome and get translated into the protein made up of amino acids. So this is gene expression. It's made up of transcription, translation. All right, how do you get them, tell them apart? DNA to RNA, RNA to protein. DNA to RNA transcription has a C in it. Transcription has a C. Translation has an L in it. C comes before L. It's alphabetized. So transcription is DNA to RNA. Translation is RNA to protein. But there are other types of RNA. We are going to learn about tRNAs. And these are going to be involved in bringing an amino acid to the ribosome and lining up with the messenger RNA. There is also an RNA called ribosomal RNA, which is part of the ribosome. And so it actually, the ribosome is made up of proteins, the ribosome structure is made up of proteins, yes, but it's also made up of a little bit of RNA. So there are, we now know that there are RNA besides just messenger RNA. So some DNA gets transcribed into RNAs and then they function, they do their job as an RNA. And this is true for transfer RNA and ribosomal RNA. Now, just a point, this figure actually has a mistake right here. Did you catch it? tRNA is this one. rRNA is this one. So ribosomal RNA has this globular kind of shape. tRNAs form this kind of T structure. Stands for transfer RNA, but it also looks in this T kind of three-dimensional structure. So that's a little mistake on that. Okay, so things we need to learn about in order to get prepared to learn about DNA replication, some things you need to know first. The key to DNA replication is that complementary base pairing. Here's my double-stranded DNA that you've gotten very familiar with. In order to copy DNA, to replicate DNA, the first thing that has to happen is these two purple strands separate. They strand separation, they pull apart. Now you've got one side with its nitrogenous bases, those orders of GCs, A's, and T's on one strand, and then the opposite, the complementary. So if it was GCAT, it's now CGTA. The complementary one is over here. Now, because you know what pairs with what, remember it only fits one way, whenever there's a G on one side, you're going to fill in with a C on the other. So when you pull these two strands apart, each one is capable of being used as a template to make the other side. And that's what we see happens. Replication, you get strand separation and the purple strands come apart, and then the enzymes are going to build based on the structure of the original strand. So purple is my original, and you see it built a brand new strand that's colored in this teal green color. This type of replication is called semi-conservative. So each new DNA, when we talk about replicating DNA, contains one original strand and then one new strand. And so that's referred to as semi-conservative replication, and it's able to happen based on that template concept, that if we know that there's a G here, we know what to put on the other side from that. And you can see the next time the cell divides, so 1 to 2, 2 to 4, you see those two pull apart and it makes a new strand for each of those. You can see how that works. All right, so you need to understand semi-conservative replication and complementary base pairing with the template concept. This is another diagram, so a little bit more detail on this. Again, remember these are all two-dimensional drawings of an actual chemical molecule. So in this case, there's my sugar phosphate backbone. We're not really paying much attention to that because we're really focusing in on the G's and C's and T's and A's, and we know how that pairs. So, so the template concept, we're going to pull these two apart, and then we're going to use that to add in the complementary nucleotide from that. So here's an A. We know we need to put a T over here. Now, what I want you to see in this figure is, remember that 5' and 3' end? You remember that 3' hydroxyl I spent so much time on in the other video, and that you can only add to that 3' hydroxyl? So the enzyme responsible for building this can only add a nucleotide to the 3' hydroxyl. So if this is the 5' end and the 3' end, we know this is the 5' end. Where's my 3' hydroxyl? Right here. Here's my 3' hydroxyl. Here is where that enzyme is able to put a new nucleotide in. So it's going to add here. If you notice, remember that anti-parallel? Here's my 3' end and my 5' end, right? Anti-parallel. There's my 3' end and my 5' end. So my complementary strand that's forming, where's my 3' hydroxyl? Right here. So that enzyme is going to add in this direction. So you notice replication is happening on both sides, but it's happening in a different direction. And it's because that enzyme is going to do its job no matter which way it's facing, up, down, side to side. And again, remember these figures can be up, down, side to side. We're always looking for that 3' hydroxyl. So don't get into top, bottom, right, left. Always look for that 3' hydroxyl, and you can figure out which direction replication is occurring. And then these you can see are the completed strands. Okay, so let's do another shorthand and something else that you should be responsible for. Sometimes you'll see DNA written out as this shorthand, and this is just representing your nitrogenous bases. We know there's a sugar phosphate. We don't need to draw it out. So this shorthand is just looking at the nitrogenous bases, which can be different. So now I'm asking you to draw the complement to this DNA. So again, humor me, take out a pen and pencil, and draw the complement to this DNA. All right, how did you do? Hopefully you knew that every time there was an A, you would put a T. Every time there was a T, you put an A, and you were able to do that. You were building DNA. We're talking about DNA replication, so we're still using thymines and not uracils. You're building a double-stranded. I'm going to want you to label things. because that 5' and 3' end is very important. So if this is the 5' end, you know that this is the 3' end of your strand. And so actually, this is the 5' end. You should be adding them this way if you're talking about which one it is, because it's always adding to that 3' hydroxyl from that. But you should be able to do this exercise. So if you need help in that, stop and get with your study group and figure out how to draw in that shorthand version. Okay, I'm going to come back, stop this video here, this introduction to DNA replication, and now the next video will get into the nitty-gritty enzymes and what's going on at the cellular level or at the molecular level as DNA gets replicated. All right, see you then.